Okay, thank you all for being here in this beginning of summer, which is always very, very difficult to stay and stay inside and listen to presentations. I hope you can see the screen now. And what I will talk to you about today are the financial and scientific challenges in the research and development of new antibiotics. It's going to be sort of an unusual presentation compared to the excellent talks that many of you may have heard during this series of the giovedì della SIMGBM. But I think it's important because my perception is that not many of us are aware of the current situation for new antibiotics. So the outline of the presentation you can see here. I will spend very little about the threat of antimicrobial resistance, and then I will focus mostly on how the industry has changed over 1 or 2 decades. What are the scientific challenges for finding new antibiotics and where we stand now in terms of incentives. And then at the very end, I'll tell you something sort of more of an adept personal experience and something about Naicons that Paolo has already anticipated. So we are all aware as member of a microbiology society of the threat that's posed by antimicrobial resistance. And we are also aware of the possible consequences. Now, but in terms of actions, what can be done? Okay, we can prevent infections, we can prevent spread of antimicrobial resistance, and we need new therapeutic options. Certainly, we will need more than one approach. Not one of them will be sufficient. But the other important message, which is, I think, is critical for understanding the current scenario, is the need for antibiotic stewardship. And I'll come back to that during the course of the presentation. So what are the financial issues? Let me start with the bottom line. The bottom line is that new antibiotics are not making money. Why? Let's look at this table, which I compiled a couple of months ago by looking what is available online. You can see a list of antibiotics: daptomycin, fidaxomicin, plazomicin, dalbavancin, oritavancin, which are in the year in which the Food and Drug Administration in the United States approved the antibiotics, and the latest year for which I could find annual sales. You can immediately see that with the exception of daptomycin, all other antibiotics, the sales volume were around $50 million per year or below. And that volume is not sufficient to pay back for the high-risk investment that's represented by bringing a new drug or a new antibiotic onto the market. I don't know if all of you are familiar how clinical trials work. I guess many of you have an idea. The main point now to emphasize here is a lengthy process. From beginning to end, you can assume it's 15 years or so. You start with a lot of compounds and come down to maybe 5 or 10 compounds that get into clinical trials. And at the end, you have one approved drug. This is on average. Different programs may have different numbers, but this is more or less the average of the industry. Now, how much does it cost to go through these programs? If you look at the same slide as before, but with some cost numbers, you can see at the very beginning the screening and evaluation and lead optimization, it will cost about $2 to $5 million per program. Then you have the formal preclinical aspects before you are allowed to enter into clinical trials and add a few more millions there. And then you have the clinical trials that there is a lot of variations and everything else, but you can assume the bill will be between $100 and $300 million. Let's not forget that these investments occur in a period of 10 to 15 years. So there are some very big challenges. One is to predict the medical needs and the market value of something that will be in the market in 10 years. The other aspect is competition, because no program is alone in this world. You'll have somebody else who is trying to do something similar and may have something better or faster, be faster in the market. And the other aspect is a heavy regulatory compliance, which adds to the cost. Why does a drug candidate fail? Well, it could be because it's no good, because it does more damage than benefits to the patient. But many times, a drug candidate fails because of lack of resources. Or it fails because in the meantime, the market has changed or competition has come up with something better. So it's the consequences of all these considerations are that bringing a drug to the market is expensive. So it will cost at least $100 million. Most drug candidates will fail on the way, and this is called the high attrition rate of losing a lot of candidates along the way. And the other aspect is market will change because competition, medical needs, and everything else. So how does the industry makes money out of these? Well, so far the system has worked under a so-called monopoly market, either because of patent protection or because of market exclusivity for new classes of drugs that have been approved. But at the same time, there is the need for pushing sales as long as this monopoly market is going on. So you need to sell a lot of it, and you need to sell it fast, because before it becomes too late. That could be because of competition from generic drugs, or because competitors come up with better drugs, or changes in market and in patients' needs, and whatever. So this is the general scenario for pharma. But what about antibiotics? Well, the current antibiotic work unless the infections are caused by resistant strains. The other thing that we know, and there is plenty of evidence, the more an antibiotic is used, the higher the frequency of resistance in a particular area. And unfortunately, Italy is, like many other Mediterranean countries, has a higher incidence of antibiotic resistance, probably because antibiotic usage is not being very strictly controlled. Now, physicians are now aware that new antibiotics should be used only in case of necessity. And this is the principle of antibiotic stewardship. If we have an antibiotic that acts on resistant strains and we keep using it, well, bacteria are smarter than us, they'll become resistant. So we need to use it sparingly. And that can be summed up in this quote I read by John Rex, was quoted about 1 year ago in Nature. Invent a bad antibiotic and nobody will use it. Invent a really good antibiotic and really no one will use it because they'll keep it as reserve just in case. And this scenario has been occurring for the past few years, and it explains why sales of new antibiotics have always been incredibly low over the past 10 years or so. You can also look at the market value of companies that are focused on antibiotics that are listed in stock exchanges. There used to be that when a company reached the market with an approved drug, the struggle was over and they were able to raise money and everything else. But if you look at the list that I compiled, you have a few companies, those marked with a star, they filed for Chapter 11. That means protection from creditors because they had more debts than they could not be paid back by the revenues. Others have somehow refocused, for example, moving into vaccine or moving into cancer, but their market capitalization value that you see in the most right column is not that great. They are well below, with the exception of Paratech, they are well below what it costs to bring an antibiotic to the market. It means their value is less than what has been spent. So the market does not believe there is a future for antibiotic companies today. But it has not always been this way. And the reason, remember, that antibiotics approved today started clinical trials at least 7 to 10 years ago. And so at that time, there was money to be made. And investors were willing to take the risk with small companies because of these considerations here, which mostly goes to if the company was able to bring a new antibiotic to the market, so have an FDA approval, usually a bigger company was interested in buying a de-risked product, so by buying the entire company, and that represented a very convenient way for the investors to exit and recover their investment. Okay, just one thing to mention quickly. The reason why antibiotics were very interesting from the clinical development point of view is that once they were at the developmental stage, so after the lead optimization, the chances of success during the clinical trial are higher because animal models are extremely predictive of efficacy in humans. So the consequence of this is the attrition rate is higher during preclinical studies because there is a very powerful filter represented by animal models, but the attrition rate at the same time is lower during clinical studies because you already know that something is very likely to work. So what I like to call the good, not so old days, when there were some really major deals that I listed here. For example, 15 years ago or so, Pfizer buying Vycuron for almost $2 billion. Vycuron had as the most valuable product was Dalbavancin, which at the time was in phase 3. In 2003, Cubist bought Optimer, which had fidaxomicin, and Trios, which had this oxazodilidinone. One approved was in phase 3, spending almost $1 billion. Probably for itself to be bought by Merck in a few years later for almost for a little over $8 billion. Polyphore was able to sign a very good deal with Roche for an antibiotic that was at the time in phase 1 clinical studies. The partnership has since been ended. And there's another example. In 2014, Actavis bought Durata, which had Dalbavancin, that at that time had already been approved. Please note the difference in value between Pfizer buying Vycuron and Actavis buying Durata for about 1/3 of the value, notwithstanding the product had already been approved. So if you're interested in knowing what's going on in the pipeline now, you can go in this Nature Review Drug Discovery article that was published a couple of years ago, and you can see how the pipeline is slowly moving. But the main issues is there are very few new chemical classes of antibiotics. And the other aspect is that most of the R&D work is performed by SMEs or academic laboratories. And this poses some challenges that I will tell you something about in the continuing part of the talk. What about Big Pharma? Well, Big Pharma, notwithstanding that most of Big Pharmas that we know today were built on antibiotics and natural products, they abandoned first natural products and they abandoned antibiotics in, I will say about 20 years ago, they started abandoning antibiotics, except for the buying frenzy of the last decade or so. So, summing up the financial challenges, I think the industry has changed dramatically over the past 20 years or so. New antibiotics are not making money, and that implies that we need new economic models to make antibiotic research and development attractive. And I'll come back to that part at the very, very end. What about the scientific issues? It's not just money. There are also scientific challenges to do, and I'll try to cover some of those. From my point of view, and this is my personal point of view, the scientific issues are, well, we have plenty of marketed antibiotics, so we have to compare anything new with what is already there. I think we realize that we lack adequate technologies, and sometimes we lack basic knowledge. And I'll try to cover some of these aspects. If you look at the market antibiotics, what has happened that many antibiotics have been used extensively in agriculture and animal husbandry for reasons that were really many times not necessary. And many times this antibiotic were the same as those used for human therapy or the same target, which implied the same resistance mechanisms. And many antibiotics that were very different from those used for humans, they've been used for agricultural purposes, for example. So there is now, if you try to repurpose them for human use, you're already facing a large resistance gene pool in the environment. And of course, many antibiotics for human therapy have been used inappropriately. Most notably, and still that way in many countries, you do not need prescription to buy an antibiotic. So, from a certain point of view, what are the mistakes of the past? Well, we had a gold mine that was represented by soil microorganisms that produce thousands and thousands of antibiotics, but that gold mine resulted in what I call genetic pollution. By releasing so many antibiotics in the environment, this created a pressure for the amplification of antimicrobial resistance gene. And with any type of pollution, it's much faster to create it than to remedy it. So now we have those pools, they are in the environment, and it will take decades before we go back to the previous antibiotic time, if we ever do. In terms of technology, I like these slides, even if it's a little old, because it shows that the productivity of antibiotics really decreased starting in the 1970s. Even if new technology were coming through, genomics, chemical library screening, a lot of other things, but not that many new classes of antibiotics were coming in. Notwithstanding, during the period, there was still heavy investment by big pharma on new technology. One example, I think this is very common, and you can find a lot of stories about it in the literature. In the 1990s, there was the belief that genomics were going to provide a sufficient number of targets, and the belief that high-throughput screening and synthetic library could provide large number of hits on any target. So the idea was that drug discovery and development was just a number game. You increase the input and you get more output. So you increase the number of targets and you get more hits. You increase the size of the libraries and you get more hits. If you get more hits, you will get more leads and you'll get more developmental candidates. Well, it was too naive. The approach turned out not to be productive, notwithstanding that many companies really invested heavily in this approach. But can new antibiotics still be discovered? To me, the answer is yes. And I did a quick search in the 3 top journals, Nature, Science, and Cell, over the past 4.5 years. And this is the list that I came up with. These are all new chemical classes of antibiotics that were reported between 2017 and 2020 in either Cell or Nature. There was no occurrence in Science I could find. You can see the antibiotic name, the activity, the source, and the institution. This is what I wanted to pay attention. The source, you can see that many of them are natural product or natural product derived or natural product inspired compounds. And in terms of the institution doing the work, with one exception, which was the arrinomycin derivatives work reported by Genentech, which is part of Roche, all the other cases were either academia or academia SME collaboration. And this is very important because it has consequences on making antibiotic pipeline sustainable from the organizational point of view. For those who are curious about structures, you can find some examples here, and since all these will be recorded, you can look at them in your spare time if you're interested in these aspects. So here the message is, yes, you can discover new antibiotics. Some of them make in the top scientific journals, but many more make in high quality journals that are not in the so-called top 3. And the lesson that we should remember is that natural products are an important source not only of antibiotics, but of many drugs approved for human use. And I liked the work that David Newman, David Craig have periodically done, updating their sort of list of the origin where human drugs come from. And you can see that synthetic drugs, the blue S here, even if this is now the major effort, still represent about 1/3 of the total number of human approved drugs in the past 35 years. But what are the scientific challenges of finding hits? Yeah, I mentioned those can be found, but it's not easy. Well, we do not have an approach that was as productive as screening soil microorganisms in the golden era of antibiotic discovery. And we have to face some facts that rational design of antibiotics is still ineffective, simply because we do not know how to turn an enzyme inhibitor into an active antibacterial agent. And the reason there are the permeability barriers and the efflux systems are the major hurdles for antibiotic activity, but are also those that we do not know enough about them. And of course, the other aspect is bacteria, including pathogens, are very diverse. They have very different mechanisms to protect themselves. And about something that we know little about, we add diversity, then it becomes sort of a nightmare. The other aspect, from the natural point of view, we have not yet found highly productive natural sources of antibiotics, which means antibiotic discovery becomes a fishing expedition. It's risky and expensive, and somebody may come up doing a lot of effort and having no results. So usually the purpose of a screening program is to find a hit, which means usually a molecule with activity on a desired target, whatever the target is meant here. I'm not going to go into the details, but with measurable penetration into a bacterial cell that not resides from current resistance mechanism and has no obvious toxicity. And then, in order to progress to becoming a lead, you need usually to establish a mechanism of action, to understand the resistance frequency, to have in vivo efficacy, and to have some idea about the pharmacokinetic properties and the route of the administration of the antibiotic. So how are hits found? Well, you need to apply a test, so a sort of test to some chemical diversity. The test can be wet lab screening, can be in silico screening, can be artificial intelligence, whatever. And then you need to put this test against some chemical diversity, which can be pure chemicals, can be extract, can be virtual molecules, or you can start from an existing molecule and try to impart properties that a molecule doesn't have. But we need to keep in mind that any approach introduces biases. The first, only a small number of tests can be applied. Whether these are molecular tests or cell tests, you have hundreds of possible targets inside a bacterial cell. You can have hundreds of or thousands of different pathogens you can use in screening. So you have to select it. And selecting what you use, you're introducing a bias. Whatever the chemical diversity you use, you're only gonna sample an infinitesimal fraction of the existing chemical diversity. And we should always remember that. It's impossible to test all the possible theoretical chemicals. And let's not forget that nature has its own biases, and we know very little about the biases of nature. Why certain microorganisms produce antibiotics and others do not, why some microorganisms produce secondary metabolites and others do not, and why, for example, some bacterial targets have been privileged by evolution many times, and you find a lot of different classes of antibiotics that act on those targets. Now, what are the issues from the organizational point of view? If most hit discovery is done by academic laboratories or by SMEs, they have some characteristics. Usually they have small research groups. They usually they know a lot about one thing. So let's all call it a single scientific specialty. And because hit finding is usually a hit or miss project, usually is not suitable for PhDs and postdocs because they need to be productive. They need to come up with theses, with publications, they need to have a career, and they cannot take easily high risk projects in which no data are really no data. So in order to complement these sort of things, SMEs or academic laboratories, they need to set up collaborations, and collaborations are not always easy to set up. There is the aspect of knowledge gap. And knowledge gap becomes especially relevant when one wants to go from past the hit stage, because you need to have people with pharmacology expertise, people who know toxicology expertise, people who know how to develop drugs. And for antibiotics, that's really challenging because most of the people with that knowledge have retired or are already dead. So it's really hard to get back that information. The other aspects that is really connected to this fact, because the need for academic laboratories and from a certain point of view also for SMEs of being productive, projects are abandoned if they are not successful. So you create a lot of things that are there that could be advanced, but maybe there are no resources, there are no skills, and so on. And as I mentioned, you need different skills to bring something past the stage of hits. So this is something of sort of a roadmap that a bunch of us worked on. It will be coming up in the next few months in Nature Review Chemistry, and shows the development pipeline for antibiotics. And we have two big gaps in funding at the very beginning and the very end. The screening and hit to lead approaches, it's really hard to find funding because it's risky and because it's difficult to come up, to be sure that you come up with the results. The central parts, there is plenty of funding opportunities. There could be more, but it's not that bad. And you can now, with the recently launched AMR fund by big pharma companies, you can almost bring molecules all the way through phase 3. But once a molecule is on the market, you need pull mechanisms to encourage revenues. And there is no such a thing at the moment. So what are the take-home messages? Well, we always need new antibiotics. We will need new chemical classes, and it's not going to be easy. And new antibiotics must be used sparingly. So antibiotic stewardship should be really important part, which means considering that R&D costs will remain high, that we need new economic models for antibiotics, push incentives to feed the pipeline, and pull incentives to make antibiotic profitable. There have been lots and lots of proposals for this system. it will take me another 15 minutes to talk about it. So for those interested, I'll be happy to share my opinion about this. But let me tell you something about personal experience, because it's a funny story, and I think it's also interesting. I was lucky enough to work on an antibiotic that made it to the market, which is Dalbavancin. You probably remember the name, which is a semi-synthetic glycopeptide with natural product produced by an actinomycete, an onomoria. It has a very contorted history, which I call the Dalbavancin migratory tour. Everything started in Gerenzano, northwest of Milan. And in 1999, the rights to the product went on the West Coast, San Francisco, Versicor. Then there was a merger between Biosearch Italia and Vicuron and moved to the East Coast of the US, in Philadelphia. Then Vicuron was acquired by Pfizer. Things moved slightly north to New York. And then Pfizer divested the rights to Dalbavancin to Durata, based in Chicago. Then Durata was bought by Allergan, based in Ireland, and Allergan last year was bought by AbbVie, based in Chicago. So this is the end of the migratory tour at the moment. With a twist of destiny, AbbVie is where I started my scientific career in industry. So it seems I can never escape from my past in all these changes. What about Naicons? Many of you know, this is just a brief reminder, we have an integrated discovery engine through which we and partners can discover a new bioactive compound for a variety of applications. I'm not going to say much now, except for saying that we were lucky enough to have lots of funding from mostly from the European Union, but also from the NIH, from the Italian Ministry of Research, from Lombardy region. We're lucky enough to have a good-sized team with many young and enthusiastic people. And the final thing I want to mention, Paolo mentioned already at the beginning, we have started an equity crowdfunding campaign to digitalize our technology platform and make it accessible to anybody who's interested. And if you would like to know more about this, please contact me, drop me a note somehow, and I'll be happy to provide you with additional details. And I will stop here and thank you everybody for your patience and your attention.